We are happy to welcome today Professor Olaf van. Please introduce yourself and your research laboratory, what research you are doing, what is the goal and the mission of your lab.
Yeah, okay, thank you. My name indeed is Olaf van Tellingen. I'm a PI at the NetherlandsCancer Institute, a dedicated cancer institute located in Amsterdam in the Netherlands. It's a combination of a cancer hospital on brain cancer, and in particular drug delivery to brain cancer and finding better treatment. So the mission of our lab actually is to improve the prospect of treatment for brain cancer patients, in particular glioblastoma, but also brain metastases are within our concept. So brain cancer is a very difficult to treat cancer, and we think that there are several factors there, but one important factor is the presence of the blood-brain barrier in the brain, which not only protects the brain, but is still able to also protect tumors growing within the brain, and certainly when they are relatively small lesions. And our work is focusing at drug delivery to the brain, so small molecule drugs. And the whole idea when I started this work, which was already decades ago, is that we actually found that there are drug transporters present in the blood-brain barrier, in particular so-called P-glycoprotein and BCRP, also called breast cancer resistance protein, that are expressed at the endothelial cells that perfuse the brain, and they efflux a lot of different drugs out of the brain, and thereby protecting the brain, which is of course a very good thing. But in case of brain diseases like tumors, but not only tumors, also other brain diseases that you would like to treat with small molecule drugs, these treatments are hindered by the presence of these APC transporters. And that is what is our main focus. So our idea is to, when we would be able to inhibit the function of these transporter proteins, we might improve the delivery of a wide range of substances, small molecules, to the brain, which would otherwise not be accessible and not be useful. So this is it in a nutshell, I would say.
Thank you so much for this. Brain cancer, it is the very deadly disease. I would say one of the deadliest disease, if not the pancreatic cancer, when it's developed very, very fast. What had been the projects in your research laboratory so far? How do you decide when you give the new project to the student or to the scientist? How these decisions are made?
Yeah. Basically, we follow three potential lines for improving treatment. One is related to the use of agents that would help the efficacy of, or to improve the efficacy of radiotherapy. Radiotherapy is still the mainstay of treatment, and we are looking into ways that we can improve the radiotherapy efficacy. One way that we do this is by a system that we call so-called mitotic enrichment, which means that we want to get more cells in the mitotic phase of the cell cycle, as this is the most vulnerable site of the cell cycle for radiation. And this has been used in the past, already had been tried, but at that time they have used compounds like vincristine, which are too toxic on its own, actually. And nowadays we have quite a number of more novel agents that can do this trick. But of course, here it also is important to realize that these compounds need to enter the brain. So in this case, we are looking for compounds that have a relatively good brain penetration, and that are able to stall cells in mitosis for a certain period of time. Let's say we give the drug at time 0, and then 8 hours later, the cells will have accumulated in mitosis, and then the radiotherapy fraction will be given. And this should be repeated every day, of course, because the standard of treatment for GBM is a 6 weeks, 5 days per week radiotherapy cycle. That is one thing. The other thing is, of course, that one of the chances, and you rightly says, it's a very deadly brain disease. And there are also many clinical trials that have been performed, which unfortunately most of them all fail. And also these trials with the so-called targeted agents, like agents that will interfere with certain proliferation pathways. And that is because it's also a complex disease which is caused by several disrupted pathways. So if you interfere with one pathway, another pathway you get rewiring of the tumor signaling, and another pathway will take over. So we are looking into combinations of treatments that may give more benefit. And of course, also in this case, we need to look at drugs that either that sufficiently bypass the blood-brain barrier in order to become active. And we are looking at combinations, for example, of PI3 kinase inhibitors, MAP kinase inhibitors, and CDK4/6 inhibitors, since these are the ones that are generally overactive in GBM. We are not yet at the stage of, let's say, personalized therapy. This is more looking at the more common denominators of what drives cancer and glioblastoma growth in the brain.
Sure.
And the third line,
No problem, go ahead.
Yeah, and the third line that we have is more recently with so-called tumor treating fields, which is a very novel treatment for glioblastoma.
I just wanted to ask you about this.
Yeah. And that is something that has attracted our attention. It's very much a black box kind of treatment, because nobody actually really understands very well what is happening. We have some ideas about this, and we are now working also to look in combinations with other agents that we think can quite dramatically improve the treatment effect of tumor treating fields. And that has to do that we think that at least a major effect of TT fields is that it causes a lot of but very subtle DNA damage during the cell cycle. And with help of some other compounds, we are able to improve, to reduce the DNA repair mechanisms and to enhance the efficacy of these compounds. So these are this is the main focus on project that we have currently running in the lab to improve potentially improve the treatment for GBM.
Sure. When you work with such small molecule compounds, right? So these are the chemical molecules. These are not biologicals. How do you work with them? Do you have the chemists that specially synthesize or produce them, or you purchase the commercial molecules? How this methodology of the drug development works with you?
Yeah. Now, we focus in particular on compounds that are either in late-stage clinical trial or are already approved in clinical trial. Mm-hmm. We don't have the ambition to, let's say, develop new molecules. I think this is a completely different set of experiments that you need. So we are looking into agents that we know that they are used in clinic. We also know, and that is important, we also keep in mind that when we are doing our studies, we do studies mostly in preclinical, in animal models, that we make sure that the exposure of the drug in the mouse is compatible to what we can achieve in patients. That's another part, of course, that many studies in mice are conducted with concentrations and or doses that are not reachable in patients. So we get a nice result in the mouse, but it does not translate to patients. And this is one of the other things that we are very cautious of, that we want to know also about the clinical pharmacokinetics of the drug, so that we can more or less try to mimic this as good as possible in our mouse models to get realistic treatments of these mice. And not, our purpose is not to treat mice. Ultimately, our purpose is to treat patients. And but we think that the mouse models can give us can be very informative, at least when we take into account that we also need to look into the pharmacological aspects and the differences and similarities between mouse models and humans.
Sure. There was recent information from the FDA that the drug can be approved without being tested on the animals, the in vitro models, the lab on a chip models, that they are enough. Did it affect somehow your research in your laboratory or in your institute? Do you also apply such tools where essentially you do the testing on the chip and on the layers of cells instead of the animals?
Yeah. Well, I am, let's say, a bit hesitant to use this in particular for treatment of brain cancer, because we know that the drug delivery to the brain is such an important factor, and that there are so many factors that also cause a reduction in the brain, in the drug delivery in the brain. So no, we are not using these techniques. I think they may be worthwhile for, let's say, tumors that are readily accessible by drugs that are systematically being given. But for our brain cancer models, we think that having access, giving mice realistic drug levels, and then actually getting tissues from these mice in order to also look at downstream effects, whether they are biological effects or biochemical effects, to confirm that the drug actually is doing what it should do, is a major necessity actually in this work. Yeah.
Sure. Speaking about the tumor treating fields, glioblastoma had been one of the best known cases, and the Novocure as a company with headquarters in Israel had been successful since multiple years. Do you see any other opportunities for any other types of the solid cancer that have very good chances for treatments with magnetic fields?
Yeah, well, they're currently, of course, they are using this in a couple, or they're trying this in a couple of other diseases. Again, here I think it's there with the tumor treating fields. There are two important points, and that's of course that the field of view of the electrical field should be well confined to the tumor, and of course for brain that is perhaps relatively simple because it's quite a fixed environment with the skull above there. For other tumor types that might be more complex, but yeah, that is one point. And I think also that we need to better understand the biology between of TT fields, and this is a very active field actually, and there are labs busy on that, similar to us. And I think we may enhance the efficacy, and I think that should also be possible. And also would be very helpful, because in its current setting, even also in GBM, the effect, of course, is still relatively subtle. Although I think it's quite an achievement that you can get this improvement in survival in GBM patients at all. Remember that temozolomide was approved in 2005, and it only gives a roughly 3-month extra benefit. So the increments in treatment efficacy in GBM are very small, and we are already happy when we are able to increase the quality, the patient's survival with a couple of months. So yeah, that makes it a kind of different field than other tumors, where, of course, like breast cancer, where survival terms of 5 to 10 years are getting more and more general.
Sure. Every type of cancer requires very different approach. Our audience can comprise from those who are interested in joining your research laboratory, whether they are students, whether they are scientists. We can also have those who are investors, whether these are business angels or VCs, as well as those who are currently affected by the glioblastoma, by brain cancer or any other type of cancer, and they are searching for information. What message would you send to these three groups of people? Those who want to join your lab, those who are patients, and those who are seeking good investments?
Yeah. Well, those who want to join my lab, I always invite them to write a letter and the clear motivation in the letter, what they would like to learn in our lab and why they would like to join. To investors is that yes, we are looking at collaborations also with commercial partners or with other partners, and I think that it's important that our research is aimed to improve the treatment of cancer patients, and we are always interested to look into novelties. Of course, we will only take on those things that we think are most promising. And to patients and others, of course, yeah, sometimes I'm also being noticed by a patient who finds my email address and asks questions. I always tell them that I'm, of course, just a doctor for mice and not so much for patients. I can help them a little bit in finding out what are the current standards. But also here I have colleagues here that are neurologists that actually treat brain cancer patients, and usually I refer them if it comes to, let's say, the treatment of their disease and what they can best do. So that is I think the most honest answer there. Yeah.
Sure. Thank you for this information. Would there be anything else you would like to add besides that what we already spoken about?
Yeah. Well, just to add again, I think that the brain is a very non-permissive site for treatment, and I think that the idea of using transport inhibitors is can serve as a kind of a platform to bring in many more compounds in the brain that would otherwise be not very useful. And I think that this is an area that is not receiving the amount of attention that it actually should have. I think many people are busy with finding novel drugs, but nobody, or not nobody, that's not true, but not too many people are actually looking into whether these are also suitable for treatment of diseases inside the central nervous system. And I think that this is one of the major focuses. It's also, as I mentioned, I work in a cancer institute, so we are working mostly on cancer, but the work that we do is more widely applicable to all kinds of brain diseases, including ALS or including Alzheimer's, etc., provided that you have a molecule that you would like to bring to the central nervous system. This might be a very generalized system to improve the drug delivery and hopefully by that also the treatment of these diseases.
Thank you so much. It was a pleasure to have you.
Good. Thank you very much. Bye-bye.